serve different functions. The brain has 10 to the 11th or 100 billion brain cells in it. These are the neurons that talk to each other and form the networks. And where these neurons talk to each other are at tiny connection points called synapses. And you have 10 to the 14th or 100 trillion synapses approximately in your brain. This is more synapses in your brain than there are stars in our galaxy. It's just a phenomenal amount of synapses. And I'll talk a lot more about those because they're very important to the disease and they're what we study in the lab. So I'll tell you more about synapses in another slide. But right now we'll zoom in a little bit on the brain. So the middle panel here is showing you what we call a coronal section through the brain. So it's cutting through the brain this way after someone has died, obviously. And we have a red box on there showing a zoom in on the neocortex. So gray matter is the stuff that's not actually gray in a fresh brain, it's sort of pink. And the white matter is on the inside. The gray matter is these ribbons along the outside of the brain where the cell bodies live. And that's called the neocortex on the outside there. And the white matter is where the wires that connect the brain cells travel through. It's white because those wires are insulated with fat, just like we insulate the wires that we use for electricity, because the neurons actually conduct electricity. They take an electrical signal from one neuron, send it down these long axons, these wires, and transmit that signal via the little synapse to another neuron. And this is how the brain's connected and wired up. These are two neurons, a red one and a green one. The cell body is the blob in the middle. That's where the nucleus and the DNA live. And the processes, they have these beautiful processes sticking out from the cell body. They have dendrites and axons. The dendrites receive signals from other neurons, and the axons send signals down to the next neuron in the circuit. And the little connection point between the two is called a synapse. And the reason I'm banging on about synapses is because these connections between brain cells are really the fundamental building blocks of learning and memory in our brains. So all of the neuron cell bodies in your brain, you pretty much had those before you were born. You get a few new ones and you lose a few during your life, but it's a fairly static population. But synapses, on the other hand, change in response to the environment. So you learn because your synaptic connections get stronger. You forget because those connections get weaker. So you gain and lose synapses through your whole life. And because synapses are so plastic and you can get new ones, they even have the potential to grow back if we can stop the damage to them. So you can see the thing I was talking about on the slides, you can see the neocortex, the curly bits on the outside of the brain. Those are the parts of the brain that are involved in memory and thinking, learning, also movement and vision. It's an amazing texture. It's sort of a cross between meat and jelly. But this is now cut in half, and you can start to see the difference between gray and white matter. So gray matter is on the outside, and like I said, in a fresh brain it's actually quite pink, it's not very gray. And if we cut in a little bit further, you'll be able to see that better. But now we have a coronal section through this pig brain. This stuff that's coming off is quite interesting. This is called dura. So your brain is protected by the blood-brain barrier, and it has a layer of meninges around it that holds in the cerebrospinal fluid, protects it from the blood, which is actually toxic to the brain. Anyway, you can now see on the inside, I'll get my forceps here, this is white, the white matter, that's the wires going through the brain, and the gray matter is where the cell bodies are going around the outside. And I'm just trying to show you here, it's a little bit small, but this little curly seahorse-shaped part of the brain right here is called the hippocampus. Hippocampus is Greek for seahorse, and the hippocampus is essential for learning and memory, so it's one of the places that is destroyed first in the process of Alzheimer's disease. We take tiny pieces of brain samples and dehydrate them through ethanols, alcohols, and then we put them in this liquid plastic that cures overnight in an oven, so it becomes a very hard plastic. It seems like a strange thing to do, but the reason we need them to be in this really hard plastic is because we need to cut them really, really thinly. So synapses are less than half a micron. So a normal microscope, even a very expensive laser microscope, doesn't have the resolution in the Z direction, that's up and down, to image a single synapse. So if we want to say, in brains of people who had dementia, what's different about those little synaptic connections, we needed a new way of looking at it. So we developed this technique, we adapted array tomography for use in human brain. And I'll show you now how we cut the sections holding water. And then there's a diamond that you can just see on the screen here. This is a 6 millimeter wide industrial diamond. It's moving every time this block, which is one of those blocks that I just showed you, every time it spins around, it's coming a tiny, tiny bit closer. It's coming 100 nanometers closer to the knife. And now it's just started cutting it, and I'm making them progressively thinner so that we can get them small enough so that they only have one layer of synapses. And we take these sections and we put them onto a microscope cover slip. So our focus in the lab is to try and understand at a fundamental level what's going wrong with these connections in the brain and to devise treatments to protect them and even to get them back. But there is a lot of hope that we'll be able to find treatments that will help